July 20th, 2021, U.S. Senator Rand Paul and Dr. Anthony Fauci got into a nasty fight on TV about research that Dr. Fauci's organization funded in Wuhan, China.
It's a dance, and you're dancing around this because you're trying to obscure responsibility.
Rand Paul claims that the research was a dangerous type of gain-of-function research that had been banned in the U.S. at that time. Fauci responded by saying,
"Senator Paul, you do not know what you are talking about."
Both of them called each other liars.
And if anybody is lying here, Senator, it is you.
Which one is right? Which one is the liar? Hello, everybody. Welcome to the live stream. That was a clip from my video from last week. I don't know how many of you all saw that. If you did, yeah, let me know down there in the chat. It's really fun to be here doing a Sunday morning science. It's been a long time since I've done one of these, so yeah, it's good to be here. I've got a presentation actually that I pre-recorded a chunk because I wanted to watch it with you guys, and I might pause it every once in a while. It's just a short, kind of a 10-minute thing that goes over the paper fairly quickly, but also it hits every little chunk of the paper that Anthony Fauci and Rand Paul were arguing about. And I wanted to pre-record that just so that I make sure that hit all the points and I don't stutter or anything like that. I just wanted to get through it really nice and clean. And then any questions that people have, I want to answer those. I'll be reading questions actually as I'm showing that little clip. And yeah, I'm very excited for this. Ah, yes. Nice intro. Yeah. So I think before I do that, however, it would be good to talk a little bit about gain-of-function research in general. And I have prepared a couple of slides. If you've been watching this channel for a while, you've probably seen some of these slides before. I did a whole video on gain-of-function research months ago. And actually, I've been trying to get people to pay attention to this since February of 2020. So for those of you who don't know, I run two YouTube channels. I run Stated Clearly, which is my main YouTube channel. I do science animations for high school and middle school over there. And then I have this channel, Stated Casually, where I do, stuff that is a little more casual. And back in, well, at the very start of the pandemic, before even most people in America started taking it seriously, I had created a video about the origin of the coronavirus, the possible origins of it. And I focused mainly on the natural origin hypothesis in that video, but I mentioned in the beginning that there are two other hypotheses, which one is that it could be a bioweapon. And at this day and age, you always have to check to make sure that that's not the case, because we do now have the technology to make viruses as weapons. So I briefly mentioned that in the animation, and then I briefly mentioned the possibility of a lab leak. And then I spent that entire rest of the video talking about the natural origin hypothesis, why it is that we would assume that a new virus would be of natural origin. But I wrote and published at that same time on the Stated Clearly website an article about all three of these possibilities, and I focused more on the lab leak hypothesis, where people were out studying viruses or collecting viruses, and someone doing that got sick, and that could be a way in which the coronavirus came to be. And when I published that video and that article, people were very upset at me for that, because at the time it was said that the lab leak hypothesis is just a conspiracy theory. I actually had teachers write to me saying that they can't use my video in the classroom because it talks briefly in the start about the fact that a virus could have come from a lab, and they said that that's a conspiracy theory. And that was kind of frustrating. And I had people write me letters as well, because I had the article on my website, on the Stated Clearly website. They were very upset that I had an article mentioning that on there. And yeah, it was frustrating to see that happen, because we don't know the origin of this virus yet. We still don't know. It's been over a year and we don't know. And we have to, the way that science works is you have hypotheses and you rule them out. You figure out what actually happened and what did not, and you check things out and you rule them off. We haven't been able to do that yet. We haven't been able to rule out a lab leak. We've pretty much ruled out the possibility of this being a intentional weapon. We don't have any reason to be concerned about that. No government official has come out and said that we should be concerned that this was an actual attack, and it doesn't really look like it was. I mean, some people are saying that it could have been something being studied under someone's bioweapons program and it leaked that way, but it wasn't. It doesn't seem like a bioweapon, and it doesn't seem like it was intentionally released. It seems like it was either an accident from nature or an accident from normal medical research that was happening. And in today's video, we're going to talk about that type of medical research that it could have been if it was a lab leak. And this is something that the Republicans have been pushing really hard that we investigate. And it's been unfortunate for me to see that this has become a bipartisan issue. It's the Republicans are all for the lab leak hypothesis and the Democrats are all for the natural origin hypothesis. And I don't understand why we got divided on political lines that way. I mean, I think historically maybe there's some reasons for that, but it's unfortunate because it's just a mystery. We don't know the origin of the current coronavirus. So let's talk about gain of function real quick here. Got my slides. Let's see. Hold on. So just so that everyone's aware, there actually is a bipartisan request right now to investigate the lab leak hypothesis. So we've got 2 Democrats and 2 Republicans that have just recently written, this was July 27th, asking for an in-depth investigation. And they're wanting, everything to be looked at. They're wanting the Wuhan Institute of Virology to be investigated further. They're wanting more information from our intelligence agencies. And they're also wanting to investigate the possibility that the NIH was helping fund not just gain of function research, because there's a very specific definition for that. And as I talked about in last week's video, Anthony Fauci says that we haven't been doing gain of function research in China. And if you look at the technical definition that the federal government uses, it's pretty much guaranteed that he's right. That definition is very specific. If you look at the more general definition that other scientists have used in the past, maybe you could say that some of what we've been funding in China is gain of function, but he was asked by federal officials if we were doing gain of function research, and he said, no, we're not doing gain of function research. And of course, because he is a federal official and other federal officials were asking him, it makes sense that he would have used the federal definition for gain of function research. So I really don't think that there's any chance that he's going to get in trouble for having said that. I know people are trying to have him arrested and saying that he lied to Congress and so on. I don't think that's going to go anywhere, because it's just, there are technical definitions, operational definitions that the government has in place, and what he said fits those definitions. But this investigation wants to go further and just look at, have we been funding anything inappropriate in China, anything that's too dangerous or too risky in the Wuhan lab. And I'm really interested in this, how this goes down. The research we're going to look at today is questionably too risky for us to have been doing. You can be the judge for that, but we'll look into this paper. It's a really interesting paper, but I am excited to see how the rest of Congress ends up feeling about all this stuff once they go through these investigations. That'll be interesting. This is what Rand Paul had said, his claim.
Viruses that in nature only infect animals were manipulated in the Wuhan lab to gain the function of infecting humans.
So that's what Rand Paul was saying. He's a little bit confused as to what actually happened there, and we'll talk about that here in a little bit. But what he said they were doing is not what they were actually doing. But what is gain of function research, anyways? And I'm going to be using more of the federal definition here than the general definition, because in general, like anytime you give an organism a new function through some sort of lab manipulation or even selective breeding, it's considered gain of function by geneticists. But in the general, day-to-day speech. But the federal government is specific about it being virology experiments that aim to increase the pathogenicity. So, how nasty is it once it gets inside of you? And the transmissibility or the immune or drug resistance of viruses with pandemic potential. So they're concerned about viruses that have pandemic potential, so they can be transmitted through the air. And they're concerned about, any experiment that intentionally aims to increase the pathogenicity and the transmissibility or the immune or drug resistance of a virus that has that potential. I was talking about this with my dad, and he said, so what you're saying is that gain of function research is when a researcher will build a monster in the lab so that they can figure out how to fight it if they ever meet it in real life. And that's a pretty good way to think about this. We build monsters in the lab. We learn how to fight them in what's supposedly a safe environment. So then when we meet them in real life, we know immediately how to take them out. So that's the idea behind all of this. But of course, whether or not that's a good idea or a bad idea depends on how safe our labs are. And these are, there have been spills from time to time over the years. So why do we do gain of function research if it's dangerous? This is a chicken farm in America, and you've probably heard me talk about this before. These are horrible animal abuses, but we're not going to talk about that today. We're going to talk about the fact that these can produce viruses, right? When you have a lot of animals that are stuck together like this, you can generate new viruses. New viruses can evolve in a scenario like this. There are lots of viruses that infect birds that can jump over into mammals, that can jump over into humans, and so on. And so this is a big problem because we are mass producing these things. They're living on top of each other. They're pooping on top of each other. And some of these chicken coops are really nasty. So this is one of the worst ones that you can find. I can't remember. This is like this is a picture I got from a website that goes and does like animal welfare stuff. They try and take pictures at the worst places they can find. And so that was interesting. But the 1918 Spanish flu, we think, was caused by a fusion of a bird flu and a swine flu that then jumped into humans. And so we know that the way that we farm animals, it's just, they're breeding grounds for new viruses. Viruses can evolve in these scenarios and jump over into humans. And we went through this 100 years ago, and we're going through it again. It's a horrible situation. And so the point of some of this gain of function research is to try and get ahead of that. So this research right here from 2012, this is the experiment that really scared everybody. This is what got everybody freaking out about this thing called gain of function research. And this is where they took a virus that has a 60% mortality rate in humans. It's more deadly than Thanos. All right. Our most evil supervillain that we can invent in Hollywood is not as evil as this virus. It will kill 60% of the people who get it. It doesn't transmit from person to person, though. That's the good news about it. But if you work with chickens and they have this virus, you can get it. If you're working with them a lot, you get a really, really high load of this in your lungs when you're working with chickens, and it takes hold on you, you have a really high chance of dying. And what these scientists did is they took this Thanos, deadlier than Thanos virus, and they gave it to ferrets. And they would squirt it in the nose of a ferret, and then they would let that ferret get infected. And then they would take the infection and squirt it in the nose of another ferret. This is called serial passage. And they would keep on doing that until it evolved the ability to pass from ferret to ferret just through sneezes and breathing. And then they studied the genome of the virus to figure out how it evolved that trait. What were the mutations that happened? And that was this paper. And they gleaned a bunch of cool information, you could say, from that. Some people argue that this doesn't really help us at all. But it was extremely risky. I mean, they took a virus that can only be transmitted through the air from bird to bird, and they made it so it can be transmitted from mammal to mammal. And we think that probably it could be transmitted from mammal to humans. I mean, we're mammals as well. And our lungs are actually very similar to ferrets. So this was absolutely terrifying. So this is not the stuff that was done in China. This was done like way before. This is the very first experiment that kicked off everyone's worries about gain of function. So this was actually done in the Netherlands, I think. And it was done in a really high-class lab. Everything was safe. Nothing bad happened, but everybody freaked out. Like, this is crazy. Why are we doing this? So here are the reasons that people have said that we should be doing gain-of-function research. These are the arguments for gain-of-function research. Gain-of-function research, this type of gain-of-function research, where we make a virus more dangerous than it was before, it lets us see how close a virus is to being able to infect humans. How far is it in its evolutionary pathway to spilling over into the human population? The other reason is so that we can find out how a virus infects humans. And, when we have the virus, a version of the virus before it can infect humans, and then another version of the virus after it can infect humans, we can see by studying its genome exactly what it took for that spillover to happen. Then we can watch how resistance evolves. So if we've got a medication that we're using to treat a virus, we can do some experiments where we let that... We treat that virus the same way we'd be treating it in a human, and we just keep on doing that over and over and over again until it evolves a resistance to that treatment, and then we see what it did to evolve that resistance. Like what mutations were there in the virus that occurred that allowed it to overcome that resistance. Another way is, this is one that I'm not so familiar with, but it's people claim that we can use gain-of-function research to learn how to make more effective vaccines, and I am not I don't know enough about vaccine production to understand why gain-of-function research helps in this scenario, but some people claim that it does. And then last but not least, to understand the genotype-phenotype link. We don't really understand, if we're just looking at a sequence of nucleotides, we can't really predict how that's going to affect a virus. And so gain-of-function research can help us learn the genotype to phenotype link. So that first, in my opinion, I'm going to give you my opinion here, that first reason for doing gain-of-function research is not at all even close to being worth it, in my opinion. Evolution is so crazy. To think that we can watch evolution happen once and then predict how it's going to happen again and how likely it is for a particular virus to jump over into humans, I just think that it's very suspect. It's not, the information we can glean from that, I don't think is worth the risk. I mean, this is an insane risk. I mean, like I said, with this, I call it the Thanos virus, H5N1, we created an absolute monster in the lab that doesn't exist in the wild. And that was very reckless, in my opinion. And it was basically to see how this would evolve and how close it is to jumping into humans. I think we should just assume if there is a virus that can infect humans sometimes in the poultry industry, that it's close to being able to evolve to transmit in a pandemic-like fashion. I think that's just what we should assume. finding out how a virus infects humans, I think that might be kind of helpful, but I highlight that in yellow here as in caution, like yellow. And then to watch how resistance evolves, again, that's that's probably helpful, but let's be really stinking careful how we're doing that, because that would be catastrophic if we unleashed a virus that's no longer treatable onto the public by accident. Just someone got sick at the lab, and that would be horrible. And then, again, learning how to make more efficient vaccines. I don't even know enough to have an opinion on whether or not I think that's a worthy use of gain-of-function research. And then to understand genotype to phenotype link, I don't think that's a worthy use of gain-of-function research, because we can study this in other ways. We don't need to create monsters to do that. So, yeah, gain-of-function research is very controversial. A lot of people say we shouldn't do it at all. I lean that way, but other people have made fairly convincing arguments for doing it. But I don't think that the labs that we're doing it in are safe enough. I think that we need to create labs where all of the employees go and live on a campus, and they work in that lab for a couple of months, and then they quarantine for several weeks, and then they come back out into civilization. None of our labs are set up like that right now. At least none of the civilian labs. Maybe some of the military labs are set up like that, but none of the civilian labs are. The civilian labs are set up so that there's a lot of safety precautions, like when you're actually working with the viruses, but then you just go home and, sleep with your wife at night and, ride the bus and get on an airplane and visit your grandma the next week. I think we need to change this. If we're going to continue gain-of-function research, the really dangerous kind, where you actually take deadly viruses and make them worse, we need to have the entire facility be a quarantine facility, and all the employees are quarantined after they finish their 2-month work period or whatever. We cannot afford to have COVID happen again because of a lab leak. This is so expensive for us. This is insane that we would do this. It's absolutely crazy. So, if you want to learn more about gain-of-function research and the kind of the drama that's happened in the past, what different government officials have thought about it in the past, these are some government documents about the debates surrounding it that happened. I also did a video about this, what is gain-of-function research, that you should definitely check out, where I went over all of these papers. And yeah, there's over 1,000 pages of stuff here. And I went through it all. Not in the video, I made like a 10-minute video about it, but it was a nightmare to get through this stuff. But it's interesting. So, anybody have any questions about gain-of-function research in general? And then I want to go and show you the video that I made, the presentation about the actual research that was done in Wuhan, China, that we funded, that some people say is gain-of-function research. Anthony Fauci says it's not. Legally, I don't think it is. But again, there's going to be an investigation about that. But I'm pretty sure Anthony Fauci is legally fine. But we will look at that research, and we'll see that even though he's legally okay, probably, that the research is a little bit sketchy. So, but before we do that, I want to see if I have any questions specifically about gain-of-function research. Aki Sci Channel asks, can we learn how that virus can infect humans with cells in petri dishes? Yes, that's what they're doing. They don't ever just infect humans with them. To see how a virus is infecting humans, we either use cells in petri dishes or we use what are called humanized mice. A humanized mouse is a mouse that has a couple of genes edited so that it expresses certain organs in a similar way that humans express them. So in the case of COVID, for all these SARS viruses, we will use a mouse that has a humanized ACE2 receptor in its lungs. So we take out the mouse's normal ACE2 receptor and we put in a human ACE2 receptor. Actually, I don't think we take out the mouses. I think we just add the humans. And that allows the virus to get inside the mouse lungs, even though it's adapted not to mice, it's adapted to humans. It can infect the mice and get them sick. And then we watch how that virus progresses in the mouse model. So that's one of the ways that we do it. And then we do it in cell cultures as well. So that's a good question. Sanjeev Sharma says, a little bit sketchy. Is that like being a little bit pregnant? Yeah, I think maybe saying a little bit sketchy is understating it. It's very scary what we do in these labs. And I mean, it's kind of just amazing that we've never had a catastrophic accident, you know? I mean, the coronavirus, again, there's still a high probability that it just came from nature. But, you know, even if it did come from a lab, it's not a very bad one compared. I mean, you know, we've got like a 1% fatality rate. I mean, there's pretty high morbidity rate. A lot of people are getting like permanent damage. I don't know if you've seen this, but they're calling it long COVID. And a lot of people who have had a really bad case of COVID, they're coming away with actual brain damage. And it's not clear if it's caused by the virus itself or if it's caused by being intubated for so long in the hospital. You're having a machine breathe for you. We do know that can cause brain damage when you go on a mechanical ventilation, respiration or ventilation. I always get those two confused. Whatever. When the machine breathes for you, it's not doing it perfectly, and you get oxygen deprivation to the brain, you get damage to the lungs, and this can cause permanent brain damage. I mean, when I say permanent, I mean people are being checked up a year later and they're still having cognitive failures. And COVID is doing this as well. And again, we're not sure if it's because of the breathing, mechanical breathing, or if it's because of the virus itself doing something to the brain. And that's not clear. But people are having serious problems because of this. It's horrible. So I don't want to downplay the current virus, but compared to some of these other things that are out there, other things that people have been playing with in the laboratory, this current virus is not very bad in comparison. Let's see. I'm looking for questions here before I do the deep dive into the paper. If you can do @stated casually -Jon Perry. I lengthened my username. I probably shouldn't have done that. But if you do that, it'll highlight your question in orange. And also, if you want to do a super chat, well, I'll definitely see your question. And I try to set it up so that when people do a super chat, it like announces their name and stuff, but I don't think that ended up working. I set it up wrong, but I'll try to announce your name myself if you send out a super chat. Okay, we got a question. There was a YouTube video about SARS-CoV-2 plus the immune system making erythrocytes more plastic, seen especially in some long COVID patients. Okay, yes. That was, I don't know if I'm saying that right, but erythrocytes. So yeah, that was, people are trying to figure out the cause of long COVID. Like, why is it causing these mental issues? And like one of the studies, I haven't read it, but I don't know if I should even say this on the internet because I haven't gone through it, but it was saying something like it was giving people a 7-point hit in IQ tests. And whether or not you consider IQ tests to be a good actual measure of intelligence, comparatively, if a before and after test is a good measure. Of course, they weren't doing a before and after. They were doing the general population and the demographics of the person that had COVID. They would have them do an IQ test after having long COVID, and then they would compare their IQ to other people of their age and whatever classification that person falls under. And they were finding that they were on average 7 points low, is what I had heard. And if that's the case, if the coronavirus is knocking 7 points off people's IQ tests, that's like aging 10 years, like that's not good. That is not good. And it'll probably come back, but it's going to come back slowly, So that is not good. How do we know we can make a viable biological weapon out of a virus? Well, we know that we've made really deadly viruses. And I guess depending on who you are and what you want to use the virus for, what you want to use the weapon for, any deadly virus is a viable weapon, right? If you're one of these kind of smaller countries that just wants to watch the world burn, and kind of like throw your dice, you might just use anything that's really deadly. That's fine. Yeah. In the colonization of North America, we actually used biological weapons. They weren't really engineered. Europeans were immune to smallpox for the most part. Native Americans were not. They just were not. And so we used a race-specific biological weapon. We would put smallpox on blankets and hand them out to Native Americans. I say we, but it wasn't my ancestors. I hope. My ancestors were around and were screwing over Native Americans, but they weren't doing that. So I'm not innocent in it. My lineage is not innocent in this, but they weren't doing that. You could actually engineer a biological weapon like that now if you knew a lot about genetics. You could engineer a racial specific virus. It's insane what is possible. It's really insane what is possible. We have such a nasty technology available to us right now. We're not using it for that. We're studying viruses to make the world better, but people could do really, really bad things with this. And here's just one of the weird dark sides of science is that any advancement that we make that brings us, that gives us more control over our environment also gives us a greater ability to use this technology for evil. All of our advances in virology, they have dual purpose use. Like you can use them for good or you can use them for evil. And I don't know, like in the future, this is going to be such a huge problem. We have to find a way to control viruses. We have to get better at vaccines. We have to get better as a civilization at taking vaccines and, finding ways to mitigate the spread of viruses once they're released. Because I think this is just going to be a thing that happens in the future, is that these are gonna break out from time to time. And I just imagine it's only going to get worse as time goes on, unfortunately. Okay, I think that's it for questions. Let's dive into the paper here. So, here is our paper. You can see here that this is the same paper that Anthony Fauci and Rand Paul have been arguing about. First off, let's look at the introduction. The paper was published in 2017. This is 2 years before the current pandemic started. And the paper, it was structured sort of poorly, so I'm going to skip around a bit, but I will cover each section of this study. It's a really interesting study, and here we're going to do a deep dive. Let's start by noting that This research was indeed funded at least in part by Anthony Fauci's group. There's the NIH right there. They're cited as one of the funders. The paper starts off by giving us some background on SARS-like viruses. In 2002, SARS-CoV-1 broke out in southern China's Guangdong province. It became an international pandemic in 2003, but was quickly stomped out, luckily. Hundreds of people died from it, but it was nothing like the current pandemic. Again, to be clear, SARS-CoV-1 is not the virus we are currently dealing with. The current virus is called SARS-CoV-2. SARS-CoV-2 is what causes COVID-19. Masked palm civets were found to be the carriers of SARS-CoV-1. These are these beautiful little sort of cat-like creatures. They're not really cats, but they kind of look like cats. People farm them for meat, and they farm them for a type of coffee called Kopi Luwak coffee. I'm probably saying that wrong, but to make the coffee, civets are put into cages like this. It's super horrible. They are force-fed coffee berries, so the bean plus the fruit around it, which they partially digest and then poop out, and apparently the coffee that's made from their turds tastes really special or something. People pay a bunch of money for Kopi Luwak coffee. Do not buy civet coffee, okay? It is mean to civets. It's torture for these animals. Seriously, they're put in cages and they're force-fed coffee beans all day long. They just eat coffee. Humans first got... I do want to point out there that I might have gone overboard on this little video talking about the coffee stuff, because actually the civets that the virus came from, they were, I think they were being used for meat. They weren't being used for the coffee production. But just when I learned about this coffee production, this was a couple of years ago. I couldn't believe that we were doing this. Like, first of all, how could the coffee be that much better than normal coffee, right? Why do you want to eat coffee that came out of an animal's butt? And then we're torturing animals so that we can have coffee that tastes slightly different. I don't think that's acceptable. Anyway. The virus probably by working on civet farms, but bats were suspected to be the original hosts. And the reason for this is that bats are loaded with coronaviruses, so much so that you can even find active viruses in bat droppings. So bats gave SARS to civets. Civets gave SARS to humans. That's what we think happened. The origin of SARS-CoV-1, so the first COVID. China and the US both wanted the original SARS-carrying bats to be found. We wanted to know where these bats lived. The logic was that if we knew where these bats were from, we could stop them from ever spreading SARS-like viruses to humans in the future. This was a preventative endeavor. We wanted to stop this from happening again. To find the original source, scientists from Wuhan's Institute of Virology started doing expeditions to bat caves all over China. In 2013, Chinese researchers isolated a virus from bat feces found in Yunnan province, quite a ways away from where SARS-CoV-1 first broke out, but they found it in bats, in bat droppings. And they named it SL-CoV-WIV1. I'll just call it WIV1 for short here in the future. So WIV1, it was isolated in the lab. They actually got it to grow in animal cells. They cultured it from bat droppings. And yeah, it was really similar to SARS-CoV-1. WIV, by the way, stands for Wuhan Institute of Virology, because it was that institute that was able to isolate this virus. From April 2011 to October 2015, Chinese researchers carried out an almost 5-year-long virus survey, funded in part by the US. The survey was done in the cave where WIV1 was found. So they found WIV1 and they're like, hey, this is crazy. Let's keep looking inside this cave and see what else we can find. Now, the US helped fund China's survey because, as mentioned before, we did not want another pandemic to come from China and then hit the US. A lot of people now are upset about this. They think that the US never should have funded virus research with China. But it seemed like a good idea at the time. Back then, things were so friendly between our two countries, especially in the scientific community, that this project apparently got full approval from the National Science Advisory Board for Biosecurity. That means that the Pentagon, the Department of Defense, decided that this was okay. They gave it their approval. I want to stop and talk there because I know I'm going to get crap for saying that. The I said that the relationship between the U.S. and China was friendly at that time, just several years ago. It, I mean, China and the U.S. we're not enemies, we're rivals, I suppose you could say. We're cooperative rivals. And so a lot of people feel like we're enemies with China. A lot of people online, you start talking about this, they're like, "Oh my gosh, China's evil," and blah blah blah. And I get that. Like, we have been, we certainly are rivals, cooperative rivals, but we are rivals. At that time, especially in the scientific community, we were very friendly with each other. Things have shut down since as soon as this stuff broke out, people started pointing fingers at each other and everything just clamped down. It's really unfortunate. There's this amazing thing that happens in science where people forget about these national issues and they just hang out with each other, they work with each other, they share with each other. And the scientists can forget how bad things might be or how competitive things actually are outside of that little scientific community. And that's why we have these oversight committees. So like the Pentagon oversees the research that we do in China surrounding viruses. They actually have a committee specifically designed to look at everything that happens and approve it. And the reason that exists is because scientists are really bad at judging the actual mood and situation, because we just like each other and we like to work with each other and we're all friends and it's all cool and I discovered this, let me share it with you. That's kind of the nature of science, and we're silly about this sometimes. But yeah, the Pentagon, they actually approved all this stuff. So a lot of people are mad at Anthony Fauci for doing this. They say it's inappropriate that he ever did this stuff with China, but he had approval. I mean, it went through all of the, it went through the Pentagon, and the Pentagon said it was okay. So maybe we should spread the blame, I guess. I mean, if you want to hate someone for approving that, there's someone at the Pentagon that's waiting for you to hate them, I guess. I couldn't figure out who that was. It's actually fairly complicated to figure out who's making these decisions. But the investigation that's going to be happening at the White House, they'll figure all that stuff out. So that stuff will be obvious here in the future. So if you're really upset that we would even do this type of research with China, well, scientists like to help each other out. They like to work together. And the Pentagon said it was okay. So what are you going to do? Hindsight is 2020. This paper is the final report on China's nearly 5-year-long study on viruses in this one particular cave. So, their results. The first thing they did was they cataloged what they found. So they went out and they sequenced the genes found in 602 bat droppings, or bat anal swabs, and 84 of those samples contained coronavirus genomes. Most of the viruses came from horseshoe bat feces samples. Some viruses came from trident bat samples, so 2 different types of bats that still do live in this cave. So once things were cataloged, they were analyzed. Those gene sequences were analyzed. Of these 84 samples, 11 were completely new coronaviruses that we never had sequenced before. And now we had their full genomes. The first big shocker from this analysis was that some of those viruses were up to 96% identical to SARS-CoV-1, much closer than any virus previously found. That was shocking. The second big shocker is that they confirmed that coronaviruses evolve really fast through a process called recombination. All viruses can evolve through point mutations, just normal, copying errors. As a virus is being replicated, small errors will crop up, point mutations. But viruses can also evolve through what are called recombination events. If 2 different virus strains happen to infect the same cell at the same time, they will produce hybrid offspring, you could call them hybrid offspring. Genes from one virus will end up getting spliced into the genome of another virus. It just happens. Looking at these sequences alone, we found signs of rampant recombination, which means that this might be what you could consider a form of sexual reproduction. Even though they're viruses, they do perform this type of something akin to sexual reproduction. Flu viruses also do this. They're really good at it. We knew that coronaviruses could do it sometimes, but we thought that maybe it was just sort of a thing that happens by accident. Well, this confirmed that actually they actively do this sort of recombination evolution. That was really interesting. So we have the cataloging, we have the genome analysis. Now the Chinese scientists wanted to engage in what is called characterization. This is where you study what a virus does and how it works. You want to look at each protein that the virus produces in its genome and then see what those proteins do. The US had already done extensive characterization with WIV1. So the Chinese researchers gave that virus to American researchers. The American researchers then took it to their really sophisticated labs, and they did all the characterization work here in the US. This time the Chinese wanted to do the characterization all on their own, and so they did. First, they took individual genes and tested those on human cells, so individual virus genes. And this is not considered to be very dangerous because they didn't take entire viruses and infect human cells with them. They reconstructed genes in bacteria, actually, and then they took those genes out of the bacteria and inserted them into human cells, HeLa cells, to see if those genes would produce the same types of proteins that cause the same types of effects as the proteins that SARS-CoV-1 produces. And they found out that they do. The proteins that these viruses code for do the same thing that the proteins that are coded for by SARS-CoV-1 do when they get inside of human cells. So that was interesting. These really are SARS-like viruses. Next, they wanted to characterize the spike proteins found in these new viruses. Cells are covered in what we call receptor proteins. There are many kinds of receptor proteins that do different things for different types of cells. Here I'm showing you a cartoon version of what is called the human ACE2 receptor. Coronaviruses have spike proteins that are shaped in a way allowing them to stick like Velcro to specific receptors on cells. The cell then swallows the virus, and the virus can start replicating itself, causing an infection. Most bat coronaviruses cannot get inside human cells because they cannot stick to the receptors on human cells. Their spike proteins are shaped in a way that they only stick to proteins found in bats. In a human body, they simply bounce around and eventually break apart. The gene sequence in the newly discovered bat viruses, however, looked similar to gene sequences found in viruses that infect humans. Alarmed by this, the Chinese scientists wanted to test the spike proteins to see if they really could work on human cells. To find out, they tried to isolate these viruses from the original fecal samples. Only one virus was successfully isolated, a virus called RS4874. They tested it in human cells and they found out, oh my gosh, they can infect human cells. This was shocking. So out of the box, there doesn't need to be any sort of evolution, nothing. One of these viruses that they found in that cave, it was already able to infect humans. That means that these bats in these caves are extremely, extremely dangerous. We are poised for another coronavirus outbreak any moment now. That's shocking. After finding that out, they wanted to test the other viruses as well to know how big of a problem that bat population really was. There are two ways to get a hold of a virus so that you can study it in cells. You can isolate a virus or you can rescue a virus. Isolation is where you just take animal cells and contaminate them with a sample that you think has a virus in it. So in this case, bat droppings. You infect the animal cells with bat droppings and you hope that some of the bat turd viruses are still active and will end up infecting those cells. And then you can study how those viruses work, right? It's a little bit more complicated than that, but basically, that's how you do it. As mentioned, that didn't really work. Only one virus was able to be isolated. The second method is called virus rescue. This is where you look at the genomes of viruses that you have sequenced. Sequencing the genome is fairly easy for us to do. You don't actually need an active virus to sequence its genomes. Even if the things are totally dead inside your bat turd, that's okay. You can still sequence their genes. we know how to do that just fine. To rescue them, you take those sequenced genomes, you take the information that you have, and you reconstruct those genomes in bacterial cells, in bacterial DNA. Special types of bacterial DNA that we call plasmids. They're small circles of DNA. Because coronavirus genomes are so large, they have to be broken into small chunks and reconstructed in multiple bacterial plasmids. Each DNA chunk must then be extracted from the bacteria and successfully inserted into a single animal cell. That animal cell must successfully accept and activate each fragment and then, build more viruses. Those viruses that the cell builds must then be able to infect other animal cells in culture. Rescuing a virus from its genome alone is very, very difficult. It's very complicated, but they felt like it was worth it. They wanted to know if these viruses really were a threat to humans. From their database, they selected 8 viruses that they thought were worth rescuing. But instead of rescuing them from scratch, they decided to make chimeras with WIV1, which is the virus that they already had in their lab. To create the chimeras, they deleted WIV1's spike protein gene, and they replaced it one by one with the spike proteins from the viruses that they wanted to rescue. Only 2 of the chimeras were rescued. The other 6 failed. Both of the chimeras that were rescued, they were able to infect human cells. This, again, is shocking. The bats in that cave are extremely dangerous to humans. They discovered that we are on the brink of another coronavirus pandemic. So there we have it. That is what we find in this paper. All right, so that's a lot, right? It's a pretty cool paper. The last bit of research where they made the chimeras, that's what Rand Paul and a bunch of other people, a lot of Republican lawmakers are saying is gain of function research. And actually, if you look in some of the scientific literature, it's called gain of function research, and other sub genres of science, even within virology. Some virologists have called that gain of function research, but according to the NIH's official definition of gain of function research, it does not qualify because they, first off, that WIV1 virus, that first virus that they were starting with, I don't know that this makes things any better, but it wasn't an animal virus that could only infect animals. It could already infect humans naturally. Like the spike proteins of WIV1, they work. They can get inside of human cells. So when Rand Paul says they were taking animal viruses that could not infect humans and then changing them so that they could infect humans, that's not correct. It was a virus that already could infect a human. Well, it could infect human cells. Let me sidetrack here a minute and point out that every time I said that they could infect humans, what I actually meant is they could infect human cells or humanized mice. We've never tried infecting a human with these, but our models don't always translate, right? So what works in a mouse doesn't necessarily work in a human, even if it's a humanized mouse. But because these experiments were just being done to characterize an unknown spike protein, it's not considered gain of function. These gain of function experiments are when you're mutating the virus to try and make it so that it can be airborne in humans or in mammals. You're trying to make it, you're intentionally trying to make it worse so that you can then, glean certain bits of information from it. That's not what they were doing here. They were simply characterizing unknown viruses. They chose to make a chimera instead of just creating each virus from scratch for some reason, and I'm not sure why. There's one of 2 reasons that they did that. One is that it was easier. That's actually probably why they did it. That it's really hard to create, to rescue a virus from scratch. You have to make all those plasmid segments, and you have to get them all to work inside of a cell. And these viruses were so close to WIV1 and most of their genome that it was almost silly to try and do it from scratch. It's just a bunch of work for no reason because they're almost identical in nucleotide sequence except for the spike protein. That's where the big differences were. So it makes sense that they would have made chimeras with WIV1 instead of starting from scratch on all 8 of those that they tried. Of course, only 2 of them ended up working, but they tried on 8. The other reason that they might have done it is because WIV1 has been shown it does infect humanized mice, but it doesn't make them very sick. So it infects them, it grows inside them, it makes them sick for a little bit. They lose like 10% of their weight, their body weight, and then they recover. And so WIV1 is kind of assumed to be a safer virus to play with than other ones. And we're not sure if it's safer because its spike protein is weaker or if its other proteins are weaker. So we're not sure if it's a spike protein or if it's the structural proteins of the virus that make it so that it's not as deadly. But for some reason, it's not as deadly in mice. So that they might have used it for that reason. They're like, oh, hey, I don't want to die at work, so I'm going to use this virus that's less dangerous, and we'll try out the spike proteins on this one instead. Those are the 2 reasons. They don't say the reason why in this paper, so we just don't know. But those are the 2 possibilities. And I don't think that creating these chimeras, the NIH's position is that creating these chimeras is not any more dangerous than just creating the viruses themselves from the genomes, or rescuing the viruses themselves from the genomes. There are people who disagree with that, though. There are people outside of NIH who disagree with that. They think that this is dangerous because you are building something that was never built in nature. But also, as we learned earlier, these things evolve through the process of recombination all the time. They're really good at it. These viruses are. So they're actually, these chimeras that we're building in the lab, they're actually being built naturally as well. So this is a really gray area, and there's I think there's room for good arguments on both sides. Anthony Fauci is probably going to get away just fine legally by saying that this does not count as gain-of-function research, but if he really cared about the spirit of honesty, testifying in the spirit of truth, not just technically being truthful, he would have pointed out that, hey, I'm using a very specific definition when I say this. What we did there was, some argue, is very risky, but NIH has decided that it was acceptable, and it was not against our rules at the time, because at that time, NIH had rules against doing gain-of-function research, but this was not included in those rules. So it's a gray area, and I'm actually really glad that Rand Paul caused all this fuss. It's unfortunate that it had to happen in the form of a witch hunt, but it is good that we now have the public talking about this because, like I said, I've been trying to get people to pay attention to actually this specific paper since February 2020. I have this on my website, on statedclearly.com, in the articles section. This paper is one of the papers I linked to in that article about the origin of the coronavirus, the possible origins of the coronavirus. So it took a crazy, screaming match on TV to get people to notice, which to me is just bizarre. For me, it's like I look at the literature. I'm like, this is crazy. People should be talking about this. Nobody cares. Then 2 guys yell at each other on TV, and all of a sudden everybody is talking about it. It's just, man, we're such a stupid species. But anyway, so that's the paper. Do let me know if you have any questions, and do send super chats, folks. I stayed up all night making this for you. Every time I watch, there's those creationists that have been making videos about me. They have their live chats, and everyone just dropping money on them every 2 seconds. It doesn't happen to me. Nobody gives money here. You guys. This is Sunday Morning Science. I should have like a break where I play hymns and like have a collection plate so that everybody knows that they can contribute through super chats to the show here. I was joking around. I should also eat crackers and drink wine, but that's a little bit too sacrilegious, I think, even for me. So let's see here. We got some questions. Quite a few of them. Let's see. Just because there are more coronaviruses that can infect human cells, does it mean they can make us sick too? What if they are benign? Yeah, that's a good point. They can infect human cells. Doesn't necessarily mean they can infect humans. To get closer to understanding if they can infect humans, you want to use a mouse model first. Oh, hey, the higher evolutionary. Oh, thank you. There's a super chat. Oh, they're coming in now. Gaurav Sharma. Thank you. Sorry, I was actually answering your question when. Oh, thanks. When doing the mouse models can tell us a little bit better as to whether or not they'd actually make a human sick. But even still, I mean, the way to really know if they get human sick is to get a human sick, and we don't do that on purpose, right? I guess unless you're creating a weapon. But let's see, another question here. If it does not intend to raise it's not gain of function. Yeah, if we're not trying to cause a problem, it's not really considered gain of function. It's considered, well, in this case, it's considered characterization. So we're trying to characterize these viruses and see what it is that they can do. Are they going to, how does each one of these proteins actually work? Look at that. The little thing that I made that thanks people on screen when they do a donation. It's actually working. It's Darwin writing on the back of Archaeopteryx that pops up when people contribute. That's cool. Okay. More questions. How does the technology used to create these chimeras work? Are the spike proteins created synthetically? So what we do is I'm not sure how we convert the digital sequence into a physical sequence in the case of DNA, because the company that I work with does this with RNA, and they do it with these artificial RNA nucleotides. The way that works is it's kind of works like just a printer that prints out a chain of RNA, and I imagine it works somewhat similar for creating these plasmids. But what you do is you take the plasmid out of your system that's coding for the protein, the spike protein, and you switch it for a bacterial plasmid that codes for the new viral protein. And so you end up creating the entire virus again from its genome, from these bacterial plasmids. So you go from the computer to the bacterial plasmids. Then you take those bacterial plasmids out of the bacteria, you put them in an animal cell. That animal cell reads those genes and builds the virus according to whatever genes are on those bacterial plasmids. And that is your new virus. And so to make the hybrid viruses, you just don't use the original spike protein plasmid. You use the new spike protein plasmid from the new virus. Or you could just, if it's only a couple of nucleotides different, if you could just genetically engineer that one plasmid there using CRISPR or whatever, and change it so that it matches the sequence of the new spike that you're trying to study. So it would be a lot easier to just switch out the spike than it would be to try and build the entire thing from scratch. Hopefully that made sense. I got a little bit rambly on that one. What evidence is it that viruses evolve 30% new genetic information in a matter of months? I don't know. I haven't seen that. So that was a question from the Higher Evolution. I haven't seen that. But I mean, viruses can evolve really rapidly, and they evolve through point mutations. These viruses have RNA genomes, which means they mutate quite rapidly just through point mutations, through copying errors, and through actually just damage from the environment. They mutate rapidly, and then they also can mutate through recombination, which is that's a way that you can kind of filter out the bad stuff and accumulate the good stuff, is by this sort of, it's like a really dumbed down version of sexual reproduction where you have gene swapping between individuals. So that's cool. Oh, so many people are helping out, and I got thank you. I mean, we've got here, let me just read off some names here. We got MI. We've got Ehondo88. We've got Nell C. I hope I'm really bad at reading names in general, and usernames are even harder. And then Michael Apple. So yes, thank you so much for the assistance here. Let's see, we've got some more questions. But Fauci is in a political situation. Rand Paul is hunting witches, and Fauci doesn't want to be burned. Yeah, I mean, I can understand why people have been frustrated with Fauci. I think overall he's been just a really steady public servant throughout his career. He's backed gain-of-function research for a long time. He's been in support of it. And a lot of people are like, oh my gosh, you're evil because you've backed gain-of-function research. Well, I mean, this is the first time in the history of our species that we've been dealing with this type of stuff. So the fact that someone may have been on the wrong, I'm pretty sure he is on the wrong side of this argument. The fact that someone spent their career on the wrong side of an argument like that, that's horrible. I mean, the consequences, if this was the result of gain-of-function research. By the way, the coronavirus, we can look at its genome and tell for sure that it was not created in this experiment that we learned about here. And both Fauci has said that, and Rand Paul has admitted that. Like, obviously, the current coronavirus did not come from this research. But it is possible that some other research that was happening at the lab ended up generating the coronavirus that we're currently dealing with, or it was just when people were out collecting viruses in the wild or handling viruses in the lab that someone got sick, right? That could have happened. And if we find out that that's the case, if we find out that there was some sort of a gain-of-function research or gain-of-function-like research that caused this, and then we see that Anthony Fauci had spent his whole career in support of gain-of-function research, you might want to bust out the pitchfork, right? But I mean, the way that I see it is it's like the reason that we want to know exactly what happened is so that we don't make a mistake in the future. It's not because we want to burn a witch, right? It's because we don't want to make this mistake in the future. I do think that Anthony has been reluctant to talk about this, overly reluctant, and I'm not sure why. Maybe it's because he's trying to cover for himself, or maybe it's because he's been trying to repair damage between the relationship between the U.S. and China. Because if there is this conflict between the U.S. and China, which there is now, a huge conflict surrounding this, they shut down. They're not sharing information with us. We're not sharing much information with them. That is really bad for getting to the root of this problem. We need to solve this problem. And if you look at what was the guy, the head of the CDC for Donald Trump. When he was the head of the CDC, he didn't talk about gain-of-function research at all. He didn't talk about the possibility of a lab leak. But once he was released from duty, he did come out. And there's an interview with him on CNN, and he says, now that I'm free, now that I'm just a public citizen, I can have an opinion about this. I do think it may have been a lab leak. Actually, he said he was quite certain it was a lab leak. But what I thought was interesting with that is that he said he was telling us that as the CDC director, he was not at liberty to say that sort of thing. Why was he not at liberty to say that? Well, because we were desperately trying to get as much information as we possibly could from Chinese officials, from Chinese researchers, and we couldn't do that if we're also trying to blame them for the virus. And so it's possible that all the stuff that people on the far right, on the right, see as Anthony Fauci covering up for China, trying to cover up his own butt for having funded research in China. Maybe that's what he was doing, or maybe he was just really trying to patch up relationships with Chinese officials and Chinese researchers. That's possible. But I do see why people would be frustrated with Anthony Fauci, because he really has not been very transparent about this stuff. Someone had to dig it out of him. They had to drag it out of him for him to start talking about it. And that's not good. You want your government officials to be transparent. You want them to level with you, as best as they can, And I don't think he's been doing that with us. And it's frustrating to see kind of the hero worship that he's gained in the media. Like you can buy Fauci underwear. There's like music videos about Fauci. It's become kind of like a hero worship thing. And we don't want to be in that position. Our leaders are our leaders. They're good at some things. They're bad at some things. We don't want to do witch hunts against them, but we also don't want to just trust them with everything that we have. We can't do that. The world's too complicated to just throw all of your love and trust at one person, right? Or all of your hate at one person. Let's see. But yeah, so yeah, Rand Paul, he's hunting witches. Fauci doesn't want to be burned. I also think that whatever Rand Paul's motives are, at the end of the day, he is highlighting a genuine problem. And that is that we do really risky work in these labs. By the way, some of this work that was done in China was only done in a biosafety level 2 lab, which is completely inappropriate. And actually, when NIH found out, they blocked funding. Like, you can't be doing that in a lab that's that unsecure. So some of this stuff was happening in places where it absolutely should not have been happening. A lot of it was done in a biosafety 4 lab in China. So the Wuhan Institute of Virology has both. They have all sorts of labs there. Some of them are biosafety 4, which is the top level. Some are biosafety 2, and so on. And a lot of this stuff was being done in situations where it should not have been done. So yeah, it was risky doing that research. So MI says, like in court, spirit has no place on the stand. Testifying is not. Scientific debate with room to explain. It's really hard to see these hearings because, like, the public just sees, like, this fight with Anthony Fauci and Rand Paul. They saw that Anthony Fauci got really emotional and Rand Paul was very just steady and in your face. And a lot of people see that as Rand Paul winning. And it doesn't really matter what the information is. People saw an emotional outburst from Anthony Fauci and they interpreted that as him being guilty of something. And it's really hard to bounce back from that. I've heard people say that at this point, Fauci's reputation is so bad that he should either step down from the public speaking, the public face of the coronavirus part is that keep the rest of his job. That's fine, but stop doing all these public appearances. Let someone else do it, or at least start adding more people to the face of COVID communication, right? Because there are a lot of people that have grown to hate him, and it doesn't matter why they've grown to hate him. The fact is that they've grown to hate him, and so it's hard for them to listen to him. It just becomes difficult. John Brummel, thank you. And so I actually do think I agree with that. I think that he probably should step out of the public light. And I don't even necessarily think that this is his fault. I think that anyone who was a public figure for COVID that has hurt so many people, hurt so many businesses, like a lot of people are going to be upset about it. And no matter who you are, no matter what you did, you're going to have done something that makes a chunk of people angry. And we just need to learn this in the future. If there's another pandemic, you need to cycle out the spokesperson. Don't listen to the same person every day. We're going to grow to hate them. Like a chunk of the public will grow to hate that person. So we need to cycle these people out. And we need to have people on both political spectrums, or from all sorts of different political spectrums, doing this science communication, because people want to believe people who they trust. People trust the people who are from their own political party, from their own lines of thinking. And we've got a problem right now where the president is a Democrat and the major speakers on public health are all Democrats or seen as being Democrats. And so the Republicans feel very unheard and very, they don't feel like they have anyone speaking to them right now. So of course they're not going to want to do what they're told. They're being told from who they see as the bad guy, and whether or not you agree with them that the Democrats are the bad guys, it doesn't matter. You have a chunk of America who thinks that there's bad guys telling them what to do. They don't want to do it, and all of us are going to suffer the consequences from them not obeying these public health measures. So we should find a way to fix this communication problem. Let's see. The pronunciation was just right. All right. Aki Side Channel asks, how did viruses evolve? I assume you're asking in the first place, like the first viruses. We don't know. Some of them might be leftovers from the RNA world. I doubt it. That's a long time to live in competition with cells and survive. Most of them are probably escaped from genomes. So different cells that have ended up escaping from genomes and become pathogenic. Some might be really degenerate cells. So we see there's a cancer in dogs, for example, that can be passed from dog to dog sexually, I think. I think it's an STD. It's a cancer STD. So it's a cancer cell that evolved in one dog. So it's a degenerated dog cell that has now evolved the ability to infect other dogs. Tasmanian devils have this too. They have a cancer that they spread by biting each other. So you can get a multi-celled organism to degenerate into a single-celled colony. So a cancer is just like a colony of single cells. That's what's happened in this dog cancer and in this Tasmanian devil cancer. And you could easily imagine, though it's not been observed, you could easily imagine that something like that could even degenerate into something as simple as a virus. So you could get this evolution through atrophy is one way, or you could get evolution of just genes inside of an organism that ends up escaping that organism and eventually becoming a virus. So there are several ways it could have happened. It's probably happened in multiple different ways. Probably different viruses have different origins. Let's see. As a student, learned about some viruses, bacteria evolving to become less deadly. Reason being, the more deadly variants don't spread as well when the host is dead or incapacitated. Is this happening? That's from Alex. That does happen usually. Right now, we're seeing that the new variant, the Delta variant, is just better at spreading. So I mean, evolution can go any which way, but in general, a virus will take over, it'll explode, it'll go through the population, a bunch of people will die, and the virus will eventually evolve to become benign. I mean, we have a bunch of coronaviruses already that are circulating in the human population. There's like 4 of them, I think. And they don't do hardly anything to us. They give us common colds, really mild colds. And we've had them as long as we have been studying viruses. We've known that those viruses have been here. They've been with us probably hundreds of thousands of years. And they are almost completely benign. Sometimes they'll kill people that are really old and sick otherwise, but they just give you a little bit of a cold. That's it. Those viruses have been really interesting, though, to study because they've told us what to expect about immunity to the current virus. In general, we're immune to coronaviruses, the older types, for just several years, and then we lose immunity. And so that probably means that we're going to need new vaccines on a kind of a regular basis until or if this virus ever becomes benign. So that kind of stinks, right? I don't know if you've all been vaccinated, but the second dose for me was quite spicy, you could say. That knocked me out for like a day and a half. So I'm not excited to get that every 2 years or it could even be quicker than every 2 years in the start as we're trying to figure out how to develop the best vaccine for it. But yeah, coronaviruses are they're pretty nasty. They're pretty hard to evade. But one of the reasons that we think that they evolve so quickly and they evade our immunity so quickly is because they're doing this recombination. Are you saying they should not have gone into the caves and studied bat viruses? Too dangerous? I wouldn't say that was too dangerous in and of itself, except for that it is dangerous. I think we just need better protocols. So maybe every time you go on these bat expeditions, these virus finding expeditions, you should be quarantined after for 2 weeks just as a safety precaution. And those samples should be taken to a biosafety level 4 lab or higher. I think that we should actually invent a higher level, biosafety 5. And at a biosafety 5 lab, everybody lives there on campus. They stay there for a month or whatever while they're working. Then they take a month off or whatever. And between working and taking time off, they are quarantined for 2 weeks. I think that's what we should do. We should develop these biosafety 5 labs where everyone who works there has to be quarantined before going home. So yeah, I think it is worth it to be doing these surveys out in the wild. Some people disagree with me. They say there's no point. They say that you should just survey people at hospitals who are sick for new viruses. I do think it's worthwhile to actually survey in the wild, but we need to do it more carefully. Am I gain of function? What is the diff if you do it naturally by hybrids selection, like farming, grains, versus when you do it in the lab on specific genes? Well, it's not really different. If we do passage experiments, so these kind of selective breeding of virus experiments to make them more dangerous, or we intentionally engineer specific parts, it's not like one is worse than the other. They're both really dangerous. So you're trying to make viruses more dangerous on purpose. That's not a safe thing to do. Fauci is shown to be corrupt. If Fauci is shown to be corrupt, then I'll stop supporting him. But if you try to do it by putting beside Rand Paul or Trump, I'm not going to find it persuasive. Yeah, I understand that. I'm not saying that Fauci is corrupt. I'm saying that I can understand why people who don't naturally want to trust him wouldn't trust him. Because, there's just if you want to hate someone, you can find reasons, right? And there's a lot of reasons right now that people have to hate their government officials. People are hurting. Their businesses are failing. Some people, their loved ones have died. They have anger, and they want to find someone to blame for that. And if they see anything that looks guilty or guilty-ish, they're going to throw that blame there. So that's what I'm saying, is that whoever's the spokesperson of this, they should just rotate out. They should take a break from it. They should let someone else come in, and we should just have a rotation of people who come in and do this, and a rotation from people of different political spectrums, different political backgrounds. Okay, politicians can manipulate people. Nerdy scientists can't deal with this manipulation. Oh, you're talking about me as the nerdy scientist? You're talking about Anthony Fauci? Fauci has done a pretty good job dealing with it. It's like he finally snapped during that one interview and got emotional, but he's done pretty good at staying calm during these things. It's constant for him. So, a username that I can't pronounce because it's a bunch of T's and H's and T's says, the hero worship makes him look untrustworthy despite it not being his actions. I agree with that. So I wasn't saying that Fauci should step down from his job. I'm saying that he should let someone else do the public speaking stuff, because he is hated. And it really doesn't matter if the people who hate him are right or wrong for hating him. It doesn't matter at all. Our neighbors, who we depend on to obey or to follow health guidelines, they need to have someone that they trust talking to them, and they don't have anyone right now. They don't. Let's see. I've been gone for a while here, so I'm just going to answer questions that I have the @ stated casually, so it highlights in orange for me. Sanjeev Sharma says, there was another article about the locals in that area having more antibodies against coronaviruses than normal. It's not as if the bats stay in the caves. Yes, that's true. I mean, anything that could have come from those caves is also it could be found in the people, around it. The weird thing about this current virus is that it showed up in Wuhan. It didn't show up in the south of China, where all these viruses are found. It showed up in Wuhan, where the virology lab is found. So that's a little bit suspicious, that maybe this came from a lab and not from the people. But you could have had someone who was sick, and the virus was transmitting, making people sick in the south, and people come to Wuhan all the time to sell food and sell goods and services and stuff. So you could have had it spreading without being noticed, because people don't go to the doctor all the time when they're sick, especially in, smaller villages in China. And then it just blew up in Wuhan. But the fact that it popped up in Wuhan and not in southern China is a little bit suspicious for the lab leak hypothesis. But yes, people are going to be getting sick from coronaviruses, and we do find that people in the south, people around these caves, that live around these caves, they have antibodies in their blood suggesting that they were hit by coronaviruses. Now, I do want to point out, though, that we have found in trying to do these antibody tests here in the U.S. that a bunch of Americans were found to have those same antibodies as well. And it's not because they were actually exposed to these SARS-like viruses. It's because they were exposed to different coronaviruses that we have, the 4 different versions of coronaviruses that already circulate through the human populations. We were getting false positives with some of those tests because of the other coronaviruses that exist. So yeah, there is evidence that people were getting sick with coronaviruses in southern China, but it's not 100% guaranteed that those were some of these SARS-like viruses. So there's that. The problem with those, Deanna says, the problem with those neighbors is that if that trusted source starts to say the same thing as the liberal politicians, then they're a traitor and not trusted anymore. We deal with these issues as they come, right? But what we see online are the two extremes, the far left and the far right. But there's a lot of people that are in the middle, and they're just a little bit suspicious of Fauci. And having someone who talks a little bit more in their language can help persuade those types of people. And I think there's a lot of those types of people. Lots of types of people who want to be able to believe in their government officials, but they just don't quite believe them. And so that's why I think at least having more voices talking about this would be important. Aki Sci Channel, the weird thing is that the Black Death, the first wave, came from Wuhan. I did not know that. Let's see. What do you tell people who say the vaccine is dangerous? Well, I mean, it's not as dangerous as the virus is. I mean, that's one thing that we know for sure. I mean, obviously, vaccines, they're not 100% safe. This is one of the problems with the way that science is communicated is a lot of times we act like people can't handle the truth, so we want to dumb it down. Vaccines can make you sick. This vaccine makes most people sick on the second dose for a day. It's making a lot of people sick for a day. So it's not like it's not a sacrifice, but what you gain from it is you gain immunity to this virus, at least for a while. And you get to help your neighbors not get sick. I feel like this is very much a call to war. My grandpa served in World War II. He fought in World War II to try and save people that were being persecuted. This was the story he was told. He was saving people that were being persecuted. He was trying to preserve the American way and all of this. He didn't know. He was a kid. I think he was 17 when he joined. His parents didn't know. They just knew what they saw on TV. We had to trust. There's a point where you have to trust and you have to participate in your community. We've been blessed to be born into the communities we're born into. We have so many opportunities and privileges living in these countries that we live in, living in the societies that we live in. This is a way for us to give back. We've been called to make a little sacrifice and get a shot. We can do it. We can take this vaccine. It's going to make us sick for a day. There is a slight chance that it can make you can have other side effects. There's Guillain-Barré and other things that can happen from vaccines. But we haven't seen high cases of this. This seems like a very safe vaccine. And it's much safer than the virus is. And you're going to help out your neighbors by taking it. You're going to help out your community. So that's what I say to people when they're worried about the vaccine. Let's see. Professor Dave made a video on anti-vaxxers, is what Aki Sci Channel is saying. Oh, some lucky viewer had no effect on the second dose. Some people are having that happen. There's no effect on the second dose. For me, it was, I was knocked out. I seriously slept an entire day. Whole day sleeping. And when I woke up, I felt like I was going to fall over. It's crazy how the vaccine expectations became of sterilizing effect. These are over expectations that scientists went along with no evidence. I'm not sure what scientists thought that there might be sterilizing effects. There was a worry that the vaccine would sterilize people. Let's see. Okay, yeah. That was a miscommunication there. Well, I'm going to wrap it up because I'm at an hour and a half now, and my voice is starting to wane. But this was a lot of fun. Should I do another one next week? I don't know what topic I should do it on. So, Nature's Compendium. I was fine after my second dose. My brother-in-law got it with me, and he was sick. Yeah, for a day. If you haven't got the vaccine yet, I recommend that on your second dose, you schedule the next day off, and you just buy some nice comfy sweatpants and just relax with your fever. I had a fever too, a slight fever, but yeah, it wasn't so bad, I mean, for me and for the vast majority of people who get it. So I would like to hear about your plans for origin of COVID investigation in the next video, please. I don't have plans for an investigation. The U.S. government has plans for an investigation. Well, they're trying to get one rolling. So we got bipartisan support now for an investigation into the origins. And I just don't know, if this did come from a lab. I think that by now we're never going to know because, they've had a year to, make it not look like it came from a lab if that's what their goal is, if their goal is to cover it up. So I don't know that we will find out. This might just be a mystery forever, which kind of sucks. But that's kind of okay in a way too, because if it's a mystery forever, we can say that, well, it may have been a meat market, the way that we treat animals, and it may have been a lab accident. And so we need to reform both of these industries. And it's true. We actually do need to reform both of those industries. We need to reform how we treat animals in factory farms and in these wildlife trades. And we need to reform how we do virology research. All of this stuff needs to be reformed anyways. So let's use this as an excuse right now to promote that reform. So I'm happy with that. That's why I am actually happy that this little drama has broken out. I'm not happy that it's turned into a witch hunt, but I am happy that people are now talking about it. The lab leak possibility. All right. Thank you, everyone. This was really fun, and I appreciate all the super chats. Yeah, it's really sorry. I shamed people a little bit, and then wow, that worked, I guess. But I just got another one from Deanna. Yes, do this every week. It's awesome. This was great. Thank you. There's a lot of work too doing that little prep video. I'm probably going to upload that video again just as a standalone. I actually spent all night doing that. I didn't go to bed yet today because I didn't plan to do that little video. And then yesterday I had the idea to do it and I started doing it and I did not give myself enough time. And so I'm going to go to bed now, actually. But thanks, everybody. That was fun. So long. Oh, yes. Hit the like, everybody, and comment.